Hey guys, so this is Lab 17 review for Histo, Microanatomy of the ear. Today we're focused on the inner ear, so primarily focused with the vestibular system, dealing with accelerations of your head, and then the cochlear system, the auditory system overall. So we'll be looking at this part right here, and then this part right here. All right, but the inner ear overall. So, as I said, really there's two systems involved in the inner ear. We have the auditory system, which is the perception of sound, and that's going to be all about the cochlea. So the spiral section here, and you can already start to see something we'll talk about more, but you have this inner membrane, kind of like a tube structure, and then you have the outer osseous portion overall. But the cochlea is for the perception of sound, hearing, and then you have the vestibular system, which is associated with balance, perception of head movement. And so we have three semicircular canals, 1, 2, and 3, and you can think X, Y, and Z. And then we have for linear motion of your head, right, thinking of pushing your neck in and out, and then like up and down. If you were to like go up and down an elevator, you have your saccule and then your utricle. All right, so vestibular system and then auditory system. All right, we're going to start with the auditory system, the cochlea. But as we said, we have this membranous, essentially tube that runs through this entire inner ear, and that is going to be filled with endolymph. And endolymph is high in potassium, low in sodium. And then we have outside of that, we have in the osseous membrane or osseous labyrinth, we have perilymph, which is the opposite, high in sodium, low in potassium. All right, but the high in potassium is done such that we can depolarize our sensory cells, which are our hair cells, which we'll get to right now. All right, but we are focused on the sensory structures of the ear in particular. So again, here we have our osseous labyrinth, so the bony labyrinth. This is filled with our perilymph. And here we have the membranous labyrinth, which you can see connects all part of the inner ear. And so we are focused on starting with the auditory system, which again is the cochlea. And the sensory portion of this is the spiral organ of Corti. And you can see that right here, this purple line that spirals throughout the cochlea. And so the way to think about this is it's basically just an entire line of hair cells. And so if you were to take the cochlea and sort of stretch it out in a straight line, it looks something like this. We'll have 3 chambers, scala vestibuli, which we'll talk about more, scala media, and then scala tympani. And you can see that our 3 middle ear bones, right, sound will come in, hit our eardrum, cause the malleus to move, the incus to move, and then the stapes will hit this oval window, transmit sound down this first osseous membrane, osseous labyrinth chamber called the scala vestibuli, and that will continue and become the scala tympani, and then that sound wave is going to exit out this way. But it's also going to cause a vibration of this middle area, and this is the scala media, and you can start to see it, but all of these tiny little dots that are going up, those are hair cells, which are our sensory cells that are going to allow us to detect sound, okay? And we'll talk more about the frequency of sound in a little bit, but the basic mechanism through which this works is these hair cells transmit an action potential after they've mechanically depolarized. And so for, whether we are in the cochlea and we're dealing with the spiral organ of Corti, again, and that's this organ, or that's this like structure we see right here that's running throughout this the cochlea, or if we're in the saccule right here and we have the macula of the saccule or the macula of the utricle, right? Those are the sensory. These all contain hair cells. Or if you're in the crista ampullaris of this semicircular duct, or if you're in the crista ampullaris of this semicircular duct, or this crista ampullaris of this semicircular duct. All right, so the idea is that movement of the endolymph is going to cause motion of the hair cells. And so in particular, each of these hair cells, basically the idea is that you have the top of all of our hair cells are embedded in a gel membrane. And so the bottom layer is going to be attached to like a basilar membrane. When sound comes in, it's going to cause, this guy to flop. Endolymph, the fluid, is going to cause motion of our hair cells relative to this gel membrane, and the name of the gel membrane changes depending on if you're in the organ of Corti for the cochlea, or if you're in the maculas of the utricle or saccule, or if you're in the crista ampullaris. But the basic premise is all the same. It's hair cells attached to a gel membrane on the top. And so motion of these hair cells, you have these little things called tip links, right? So those little proteins that are mechanically connected to these mechanically gated ion channels. And so when our hair cells move, the tip links are going to cause the mechanically gated ion channels to open. And then we said that they're all surrounded by endolymph. And so that endolymph with high potassium is going to go into the cell and cause a depolarization, and then you'll get an action potential into our nerves. All right, and so here's just another example of this. We said that the hair cells, top of it is embedded in a gel membrane. The name of that depends on, where exactly you are, but for the cochlea, it's the tectoral membrane. Okay, so hair cell, tectoral membrane, motion of the hair cell relative to the gel membrane and relative to the bottom layer is going to cause endolymph to go into the hair cell and depolarize it, and then you get your action potential, a signal is created. Okay, so then, just more on sensory structures of the ear. We already started to say this, but this gel membrane that's over the top of each of these hair cells, depending on if you're here in the saccule, here in the utricle, or here in the crista ampullaris of the semicircular ducts, you're going to have a gel membrane over them. If you're in the organ of Corti, that's called the tectoral membrane. If you're in the crista ampullaris of the semicircular ducts, they'll be called the cupula. And then if you're in the macula of the utricle or the macula of the saccule, that's going to be the otolithic membrane. We'll talk more about that. All right, but basic premise is organ of Corti is all about detection of hearing. The macula and the macula of utricle, macula of the saccule is all about linear acceleration, right? So that'd be moving your head in and out like a weird dance move, or moving up and down like that. And then the crista ampullaris for these, think X, Y, Z, right? There's 3 axes here, 1, 2, 3. And that would be like, nodding your head up and down, right? Forward and back. Nodding your head is really kind of like rolling your head forward towards your chest and then back. And then moving your head from side to side, right? And then the third would be like sort of twisting your head as if you're cracking your neck. Okay. All right, but we're focusing on this. So let's look at the histoslides of the cochlea. All right, so this is the histoslide we were given in lab. What in the world is so bizarre, right? I mean, so how did we get this? Well, remember histoslide's a cross section, so it's actually something like this. So here's our cochlea, here's our vestibular system, and so you can see semicircular canals, and this plane right here is our cross section. So you can see we're clearly going to bisect the cochlea, and so that's what we see here. This is the cochlea. And then here, well, here we have our saccule. Here's going to be our utricle. And then here we have one of the swellings of one of our semicircular canals. So, semicircular duct. And so that we have here, saccule. Here we have utricle, and then here we have crista ampullaris of our semicircular duct. All right, so that's what we're looking at overall. All right, so starting with, we said we have the osseous labyrinth, so that means that there's osseous is bone. So the first thing to know is that we have, so labeled 1 right here, this whole portion right here, that you can see sort of spiraling around. This is all going to be bone. So this is our bony axis, and it's called the modalis. Right, so you can see a little bit better here. Right, this is all bone. Inside of the bone, we have nerve cells, spiral ganglion cells, and those are going to send out dendrites to our hair cells right here. Okay. All right, and then those will eventually send out themselves axons as part of the vestibular cochlear cranial nerve overall. All right, but we have the modalis, the bone. Inside of that, we have our ganglion nerves, which connect to our hair cells, which are going to be here. We'll see in just a bit. All right, so then we have our 3 tubular compartments. And when we started to talk about this, we said that the bones of the middle ear will hit the oval window, and that will be transferred into this osseous compartment, osseous labyrinth compartment, which we call the scala vestibuli. All right, so scala vestibuli right here. So we're seeing a cross section through this tube, right? And so at the very top, we have the scala vestibuli. That will continue, and at the apex, which we call the helicotrema, the apex is the helicotrema, the scala vestibuli meets up with the scala tympani, so that's this guy right here. And then where all the action happens is the scala media. And so the scala media is going to have the organ of Corti, which we said was this guy right here, where we had the gel membrane on the top, and we have a basilar membrane, and in between we have our hair cells. All right, so that's going to be this guy. This is our organ of Corti. Okay, so scala media is where all the action is going to be happening. All right, so let's take a look at our scala media then. So again, we are looking at a cross-section of our cochlea. And so if you were to slice one of these tubes right here, then you would see that we have, 3 compartments, these 2 osseous labyrinth compartments filled with paralymph, right, high in sodium, low in potassium. And then here we have our endolymph. All right, so scala vestibuli, scala media, scala tympani. Okay, so then what are the actual parts? Because we care about what's going on here. So to start to get into that, first thing, we're just going to start very basic. So we have the scala vestibuli. What separates the scala vestibuli from the scala media is this vestibular membrane. And that is just, as we said, separates the 2 compartments. Then In our scala media, we have the organ of Corti, which contains our hair cells, which are right here, but we'll talk about those in a minute. Then we have, adjacent to that, we have the stria vascularis right here, and this secretes endolymph. All right, so this right here. And then deep to that, we have the spiral ligament. So this region right here is the spiral ligament, and you can see it is connective tissue, dense connective tissue that supports the stria vascularis, and the stria vascularis again is secreting our endolymph. Then we have, started to describe what's going on with the hair cells, but here we have our tectoral membrane, which is our gel layer. And you can see that the hair cells are meeting up with the gel layer. Then we have the spiral limbus, and these spiral limbus cells produce the tectoral membrane. Then we have part of the modalis coming in, and this is the osseous spiral lamina. All right, so some of the modalis coming in, the bony portion of the cochlea. All right, and then here you can see dendrites coming in from the ganglion cells to meet up with our hair cells. All right, and then we have our basilar membrane right here, which we'll talk about a little bit as we talk about the vibrations of the cochlea. Okay, so a little bit more on this. So we have our cochlea. The sensory part is the organ of Corti, which has our hair cells sandwiched between the gel membrane and our basilar membrane. And so basically the idea is that if you think of the organ of Corti, this whole part right here, right, as kind of like a harp, the idea is that as sound comes in, if you think from here, like imagine from here to here, that would be like this section of the harp. And if you were to pluck on this section, it'd be a high pitch or high frequency, right? That would be like going ee, like a really high sound. And then if you were to instead use a really long string, then say like this string, and you were to pluck it, that would be a really low frequency, right? And that would be a sound like deep sound, right? So the idea is that this portion of the organ of Corti is high frequency. This portion right here is very low frequency. And so you go from 20 hertz to, or 20,000 hertz to 20 hertz. And human speech is, I think it's, I don't want to tell you the wrong thing, but it's more towards the front higher frequencies of sound. Okay, but as you can see, people tend to lose hearing of higher frequencies. Why? Because it's closer to the origin of the sound, right? If sound is coming in through this oval window right here, these are the cells that are closer, more likely to get damaged. So you lose higher frequency first, okay? But then how does sound actually get transmitted? Like what's the movement of things? We'll talk about that a little bit more, but basically as sound comes in, you have this basilar membrane right here, and the basilar membrane is floppy, okay? And then the tectoral membrane is going to be very rigid, and there's also something called the reticular lamina, which is rigid. And so you have your hair cells right here, which are going to be organized through the reticular lamina, which is created by these supporting cells right here, which are phalangeal cells. And so the hair cells are going to extend their cilia up through this reticular lamina, which is made by the phalangeal cells, which you can see here, the supporting cells, and then attached to the tectoral membrane. Reticular lamina is rigid, tectoral membrane is relatively rigid. This part down here is floppy, the basilar membrane is floppy, and so floppy and two rigids, well, you're going to get a movement of the hair cells relative to the rigid part, and therefore when sound is transmitted, and therefore you'll have an action potential. All right, so just to recognize some cells, our hair cells are right here. Phalangeal cells are right below them. They're supporting cells. Here we have something called the inner tunnel, and below that we have pillar cells, and the function of those I believe we have not covered in the lecture. Here's inner hair cell, so this direction is inner, and this direction is outer. Okay, we already talked about the spiral limbus. We said it produces the tectoral membrane. And then this region right here are dendrites from the ganglion cells, which are going to be inside of the modalis, the bony part of our cochlea. Okay, so not too bad. I'm sorry this diagram is like really blurry, but here you can see this is the inner tunnel, and we have pillar cells at the bottom, inner hair cell, outer hair cells. These are phalangeal cells. Okay. All right. So moving on. So more on basically we said we have our phalangeal cells. And so the idea is that the phalangeal cells are supporting cells. And then we have our pillar cells right here, also supporting cells. So our phalangeal cells are below the hair cells, but they're going to extend processes up to make this reticular lamina, and then the stereocilia from the hair cells are going to extend through that into the tectoral membrane. All right, and so you can see that a little bit better here. Here's our hair cells. Here are our phalangeal cells, and you can see the phalangeal cells are sending up these extensions that are creating this reticular lamina plate, and then you can see that through that you have hair cells, stereocilia coming up, and then they're going to meet with the gel membrane that's above it. So the gel membrane will be up here. All right, so not too bad. Here's other supporting cells, but what do we take away? So tectoral membrane, reticula lamina, these are both rigid. Basilar membrane, floppy, hair cells, phalangeal cells. Phalangeal cells are responsible for making this guy. When sound is being transmitted, this guy wiggles. These guys do not wiggle. It causes hair cells, stereocilia, which goes through this guy into this guy, to move relative to this bottom part, causing an action potential. Okay, so not too bad. Basically, this page is saying what I just said, but so compressional sound waves generated by the stapes. All right, so Mellitus incus stapes goes through the round window into the scala vestibuli, then through the helicotrema, which we know is aka the peak right there, and then into the scala tympani, and then into the round window, back out. And shifting of these two rigid plates, the tectoral membrane and the reticular lamina, deviates the stereocilia, causing a action potential to occur. We know that the basilar membrane is the floppy one that's wiggly. All right, and then the hair cells are synapsing with the ganglia, or with the dendrites from the ganglion cells. Okay, and you can see that here. These are the afferent nerve fibers from the ganglion cells. We have our basilar membrane, you have your tectoral membrane, you have the vestibular membrane separating scala vestibuli from scala media. You have your striae vascularis, which is producing our endolymph, and then you have your spiral limbus right here, which produces the tectoral membrane. And then you have your osseous spiral lamina, which is this, but this is really just part of the modalis. Okay. All right, so that's it. Let's move on to the vestibular system. So the hard part is over, cochlea. And now we are dealing with the saccule and the utricle. All right, how do you remember that? Well, saccule looks a tiny bit like a little sac. And we are focused on same basic premise, hair cells extending up into a gel membrane. The gel membrane is going to look slightly different for depending on where we are. The nice thing is the saccule and the utricle, both associated with linear acceleration, right, like this, and then up and down, are going to be maculas, and they look very similar. And then the only ones different are these three crista ambularis are going to look a little bit different. But this one's nice to remember because doesn't this kind of look like a head on top of a neck? Yeah, it does. And so how do we remember what the crista ambularis does? Well, you're nodding your head and then moving your head back and forth like this. So nodding head and then, sort of twisting your head like maybe an owl to crack it. All right, so starting with the macula of the utricle and saccule. These are pretty nice because it is relatively straightforward. So that would be located right here. This is the utricle, and then over here is the saccule. And again, hair cell with a gel membrane over the top. In this case, the gel membrane is a little bit stranger. So we have our hair cells more apical, and then we have supporting cells a little bit towards the bottom. And it can be kind of hard to distinguish, but the hair cell is a little bit further up. So hair cell, supporting cells. They extend their stereocilia into a gel membrane, which is the otolithic membrane. And so why is it called otolithic? Because it has otoliths, which are calcium carbonate stones in them. And so the idea is that when you move your head like forward and back in a linear motion or up and down, these stones are going to wiggle as a result of the endolymph moving past them, and it will cause depolarization overall. All right, but it has these little stones called otoliths. All right, so again, this region right here, utricle, saccule. All right, saccule is closer to the cochlea. All right, so here we have our otolithic membrane, aka the gel layer. Here you can see our nice otoliths, or calcium stones, calcium carbonate stones. Hair cells are more apical, and then supporting cells are towards the bottom. And then here we have loose connective tissue. This is part of the osseous labyrinth. And so this is going to be full of paralymph, and here it's going to be endolymph. All right, endolymph again is high in potassium for depolarization. So this is the zoomed out version of this whole thing of a macula. And you can see you have your otoliths. This is your otolithic membrane. You have your hair cells, supporting cells, and then this guy back here is part of the osseous labyrinth. All right, this is just slightly, this section right here is the saccula, but you can see it's exactly the same. The otoliths are a little bit harder to see here, but you can see otolithic membrane, hair cells, and then supporting cells. And then here we have osseous membrane with our paralymph, and this is loose connective tissue. Okay, that brings us to our crista ambularis. Crista ambularis is really nice because, again, doesn't it kind of look like a head? It does. And so each of the three semicircular ducts are going to correspond to, right, this is nodding. This is like saying no, right, shaking your head. And this is like cracking your neck. And so basically the same premise applies. You have your hair cells more apically. Then you have your supporting cells. They extend their stereocilia into a gel layer, and the gel layer is called the cupula in this case, or cap, and it looks kind of like this. So this would be the cupula. These are our hair cells overall, and they have the nerve fibers going down, and then these are the supporting cells. And yeah, so you have endolymph, and other than that, it's pretty straightforward. So supporting cells, hair cells, this is the cupula, right, our gel membrane, and then this right here, osseous, this is paralymph overall. So not too bad. Let's do a quick review. All right, so here, organ of Corti, which is this whole thing. So that means this must be scala media. Then we have tectorial membrane. This is our the rigid lamina, and I'm blanking, reticular lamina. And then you have your, yep, reticular lamina is correct. That's 2 right here. You have osseous spiral lamina, which is just continuation of the malleolus. You have spiral limbus, which is producing the tectorial membrane, which is right here. Then you have your hair cells, right? These are outer hair cells. This is inner hair cell. These are outer phalangeal cells. This is inner phalangeal cells. This is pillar cells, pillar cells. This is our inner tunnel. And here's our basilar membrane, which is our wiggly part. And I think that's it. And then here's our afferent dendrites from our ganglion cells. All right, we already saw this guy, but this is our reticular lamina made by our phalangeal cells, the supporting cells. Here we have our hair cells, and this is our stereocilia. All right, and we already saw this. So good job, guys. That's it. And, keep at it. Don't stress yourself out too much. You've taken hundreds of exams, and, be nice to yourself. All right, bye, guys.